السلام عليكم بنات، صوتي واضح؟ أعتقد النتورك عندي شوي ضعيفة، let me know if it's not clear. طيب, without further ado, I'd like to start with our session today about lipoprotein transport. The topic is not fully or completely new for you, so I'll try my best not to make it repetitive. Right. So apparently you know quite a lot about lipids. I'm not going to talk much about lipids being insoluble, the biggest source of stored energy in the body, and the other physiological functions they serve in terms of membrane structures, precursor for vitamins and hormones, as well as bile acids. We have the different classes of lipids: cholesterol, triglycerides, or triacylglycerol, fatty acids, and what functions they serve. And we have talked quite a lot about fatty acids yesterday. This is just introductory, and it's all knowledge that you already have. So let's start talk about the digestion of triglycerides as well. This is not new. It starts in the small intestine by the action of pancreatic lipase, releasing two fatty acids and leaving one attached to the glycerol backbone. So we have monoacyl glycerol and two fatty acids. This is a hydrolysis step. Then these products are solubilized by the detergent action of bile acids and then transported from the intestinal lumen to the intestinal wall, where toward the excuse me, the cell surface of the intestinal cells. Then they are being taken up into the intestinal cells, the epithelial cells of the intestinal wall, in the format of two fatty acids and monoacyl glycerides. And then within the epithelial cells of the intestine, they are reassembled again into triacylglycerols and then packaged into chylomicrons in order to be exercised out of the intestine into the lymphatic system and in the bloodstream, where it can circulate and reach the target tissues where these triacylglycerols are needed. Excuse me. طيب, we talked about chylomicrons. Why is it needed? What does it do? This chylomicron particle. Why were these dietary triacylglycerols? Why were they needed to be assembled into chylomicron, a lipoprotein particle? So, as you know, obviously, lipids are hydrophobic compounds, water insoluble, so they need ways to be transported into the circulation, into the blood, the aqueous environment of the blood. That's why they need to be combined with water-soluble compounds, such as phospholipids and proteins, to make them into these special particles which have a hydrophobic core. So all the hydrophobic compounds, glycerol esters and triacylglycerols, are clustered in the center of the lipoprotein particle, and they are surrounded by the hydrophilic coat at the outside, which is made up of the hydrophilic substances, the proteins, the phospholipids, and the non-esterified cholesterol. So that's why they are called lipoproteins. So they are particles made up of lipids and proteins in order to transport the hydrophobic lipids in the circulation. So the protein component free of the lipids, the protein component of the lipoprotein without the lipids, is what we refer to as apolipoproteins. And they serve multiple important functions in the body, in the lipoprotein particle. First, they maintain the integrity, the structural integrity of these particles, the lipoproteins. Also, they are important activators of some activities involved in lipoprotein and lipid metabolism, such as lipoprotein lipase and LCAT. We will get into these in a minute. They are also an important sort of cell surface recognition particles, allow recognition of by cell surface receptors. By now, you must have learned at least about how many types of lipases. We had the hormone sensitive lipase yesterday, which breaks down triacylglycerides in adipocytes. We have just mentioned the pancreatic lipase in the small intestine, involved in the digestion of dietary triacylglycerides. And then we have just talked about lipoprotein lipase, and we'll see what it does in a bit. So these are at least three types of lipases that you have learned thus far, and you need to be able to differentiate between them. So let's talk about the function of lipoprotein lipase. As the name indicates, it hydrolyzes or breaks down the triacylglycerols and the chylomicron particles, and then other lipoproteins, from as the name indicates, obviously. So it's involved in the release and breakdown of triacylglycerols and the lipoprotein particles. Where is it present? Near the tissues which need to utilize these triacylglycerols, but exactly secreted from these tissues, اللي هم الadipocytes والmuscle cells, and they are attached to the endothelial cells of the capillaries that surround these organs. عشان كده نسميها endothelial or capillary lipoprotein lipases. So they have easy access to the circulating lipoproteins in the blood, where they can hydrolyze the triacylglycerols to be taken up by the peripheral tissues, adipocytes, and muscle cells. So the classes of lipoproteins, I'm not gonna spend much on that because we have learned it quite a lot in the past, but you know there is the chylomicron, the largest particle. It's two. I'll try to phrase it into a different way. Maybe it will make it simple for you to memorize. We have the two lipoproteins which transport lipids. One transports dietary lipids, mostly triglycerides, اللي هو الchylomicron, and one transports endogenous lipids, mostly triglycerides. يعني نعتبرهم الاثنين triglyceride transporters, one for the dietary or exogenous, that's chylomicron, and one for the endogenous liver synthesized triglycerides, that is VLDL, very low density lipoprotein. عشان كده when you look at their characteristic apolipoprotein particle, they are all ApoB. For chylomicron, the unique lipoprotein is B48, and for VLDL, the unique characteristic apolipoprotein is B100. The other two lipoprotein particles are cholesterol transporters. One of them is transporter or carrier of bad cholesterol, LDL, and the other one, HDL, is a transporter of the good cholesterol, or it's a good cholesterol transporter, and it's HDL. فهذول الاثنين are cholesterol transporters. One is bad cholesterol transporter because transports cholesterol from liver to peripheral tissues, LDL. The other one, HDL, is a good cholesterol carrier because it collects or transports cholesterol from peripheral tissues back to the liver. Okay, this is an easy way to memorize it. The IDL or intermediate density lipoprotein is sort of a VLDL remnant particle. As VLDL is being metabolized in the circulation, it's converted to the intermediate density lipoprotein and then to the low density lipoprotein. So sort of a transition from this density to this density particles. عشان كده الثلاثة هذولي أكيد they will still retain the ApoB100 لأنهم derived from VLDL. خلاص. High density lipoprotein, the characteristic apolipoprotein is A1, ApoA1. But it also carries other apolipoproteins for other functions. We will come across it in a bit. طيب, as you may know already, the highest the lipid content of a lipoprotein particle, the largest the size and the lowest density is. وكل ما زاد البروتين content يقل السايز وتزيد الدensity. Okay, so let's move on. This is the lipoprotein metabolism. There are two pathways, the exogenous and the endogenous pathway of lipoprotein or lipid metabolism in the body. Exogenous deals with the dietary lipids, and the endogenous obviously deals with the endogenous lipids that are synthesized in the liver mostly. طيب, so there are two enzymes involved in these pathways اللي هم ذكرناهم LPL, lipoprotein lipase, and LCAT, lecithin cholesterol side transferase. الشيء الثاني, pathways, even though they seem separate, one endogenous and one exogenous, they are interconnected. There is some sort of relationship between the two pathways, and they are all centered around the liver, as you can see. We'll break them down in a bit. Tell me whenever the voice disconnects because I can see my network is getting weak. طيب. The exogenous pathway, it handles the dietary lipids, cholesterol and triglycerides, which, as we have seen in the digestion slide, are assembled into chylomicron. Chylomicron is released into the circulation. It has its ApoB48. Once in the circulation, it will need two apolipoproteins اللي هما C2 و E. It will take it from HDL. So HDL serves an important function here is that it supplements or provides these two apolipoproteins to other lipoprotein particles. And we will see their functions in a bit. Chylomicron now it will move in the blood for the lipids content to be utilized by other organs. So it will be acted upon by the endothelial lipoprotein lipase, releasing triglycerides to be utilized. So eventually we will be left with the remnant, chylomicron remnant. So now after the release of lipoprotein, for triacylglycerols, there is no need for apolipoprotein C, because we mentioned it activates LPL. So after the removal of triacylglycerols, we don't need ApoC2, so it can return to HDL. Now the chylomicron remnant is no longer needed, so it can go to the liver where it is endocytosed and the remaining contents can be processed in the liver. How does it get recognized and bind to the cell surface receptors in the liver? ApoE. That's why it takes ApoE from the HDL particles. The endogenous pathway deals with the lipids that are synthesized endogenously in the liver. So they are assembled initially in the liver in the form of VLDL, which is released in the circulation. It has its unique ApoB100, but again, it takes ApoC2 and ApoE from HDL because they are needed. Once this particle VLDL is in the circulation, it will need these two apolipoproteins provided by HDL. So it will start circulating, and LPL will act on it to release the triacylglycerols. So it will become the triacylglycerol content will become less and less. So we will have the transition into IDL, intermediate density. It will become slightly more dense because we have lost some lipids, and then it will become LDL, more denser because again we have lost quite a lot of triacylglycerol. So LDL has more cholesterol content than triacylglycerol. Again, whatever remains from IDL or LDL goes to the liver to be utilized and processed their contents. طيب, let's start with the chylomicron a bit in details. As we said, it is synthesized in the small intestine to deal with or to carry dietary lipids. One important thing that we have just mentioned so far is how it is assembled and how it's loaded with lipids. These lipoproteins are initially synthesized as protein, especially chylomicron. It's sort of an empty particle. It has only the ApoB48. Then in the digestive tract, it's loaded with the dietary lipids. What catalyzes this transfer or loading of the ApoB48 with the lipids? MTP, microsomal triglyceride transfer protein. And there are some clinical implications that has to do with this protein. We'll see them later on. طبعا the remnants, as we said, they are removed by the liver, and being the largest particle of the lowest density, provided its function that it's 90% lipids, mostly triacylglycerides, we would imagine that it's the largest and the lowest density lipoprotein particle. تكلمنا عن الأبوبروتينز حقت الأبولايبوبروتينز ApoB48 unique to chylomicron, ApoC2 and E are provided by HDL. طيب. How about VLDL and HDL? VLDL synthesized in the liver, the function we know about it. So it's mostly triacylglycerides from the liver and some small amount of protein. The unique apolipoprotein is B100, and again C2 and E are provided by HDL. How about HDL? HDL is synthesized in two places, the liver and the small intestine. Its function is the reverse cholesterol transport. Why we call it reverse? Because mostly The transport of lipids in the body is from the liver and the small intestine to other tissues. This HDL does the opposite, the reverse, where it transports or takes cholesterol from peripheral tissues and returns it to the liver. So it's mostly protein because it's synthesized as an empty particle which functions to go around the body and collect cholesterol. عشان كده يكون sort of disc-shaped empty particle that is mostly protein, the apolipoprotein of A1, then it circulates and starts collecting cholesterol esters and increases in size and shape. It has, as we said, ApoA1, which functions as an LCAT activator. LCAT is the enzyme that forms cholesterol esters from free cholesterol. And that's important for HDL while it's assembled and carries or collects cholesterol from the peripheral tissues. It has ApoC2 and ApoE. Why? To give it to other lipoprotein particles in the circulation. So there is the VLDL pathway synthesized in the liver, initially as a nascent VLDL, similar to chylomicron. It's synthesized as nascent. Once they are released in the circulation, اتكلمنا هذول الاثنين اللي ينقلون الترايسايل glycerols, one for endogenous, one for exogenous. في quite a lot of similarities between them in the pathway. عشان كذا we try to compare and contrast in order to make it easier for you to memorize rather than or to understand rather than having to memorize. فهما الاثنين they are synthesized as nascent particles, then released in the circulation. In the circulation, they pick up the two ApoC2 and ApoE from HDL, then they become the mature particles, chylomicron and VLDL. بعد كده they return the ApoC2 to the HDL after the release of the triacyl glycerols. Chylomicron need to retain ApoE. هذا يمكن فرق بسيط. Needs to retain ApoE for receptor binding at in the liver, whereas the LDL remnant اللي هو IDL does not need to retain اللي هو ApoE. It will return it to HDL because it has its own receptor recognition apoprotein اللي هو ApoB100. طيب one important aspect about IDL for chylomicron نقول انهم similar الchylomicron والVLDL عندهم remnants كلهم والremnants ترجع بالليفر. Chylomicron remnant its name is chylomicron remnant and it binds to the liver by ApoE عشان كذا يحتفظ بالApoE. ال VLDL remnants is IDL, the intermediate density, and it has its B100, which can be recognized and bind to the liver receptors. ونفس الريسبتورز ترى اسمها يمكن شفتها بالسلايد اللي راحت ApoB B100/ApoE receptors. It recognizes both. خلاص. طيب. As for the function of LDL being a cholesterol transporter to the other tissues, we need to have more control over the process of cholesterol uptake into the tissues. So once there is enough cholesterol within the cell, we need to stop the uptake of cholesterol from LDL that is bound to the LDL receptor on the cell surface. So the increased uptake will trigger few changes in the cell that will stop the uptake and deal with the intracellular cholesterol. It will downregulate cholesterol synthesis because we already have enough cholesterol coming from the outside. So it will inhibit HNG-CoA reductase, which we know is the enzyme for cholesterol synthesis. So downregulate and inhibit cholesterol synthesis. Another thing, it will stimulate increased cholesterol storage within the cell. So this cholesterol within the cell will be stored because it's not needed. And that's through the activation of an enzyme that forms cholesterol esters in the cell, which is called a CAT, acyl-CoA cholesterol acyl transferase, transfers an acyl group from acyl-CoA to cholesterol to form cholesterol esters that are stored. The third mechanism is to downregulate these LDL receptors on the cell surface to stop the further uptake of LDL cholesterol. And this is how the body or the cells control the amount of cholesterol taken up from the circulation into the tissues. Let's move on to HDL, our good cholesterol carrier. It's synthesized, as we said, in the liver and in the intestine as an empty particle that's ready to embark and circulate to collect cholesterol and return it to the liver. So it will be an empty particle, disc shaped. It has only proteins اللي هم ApoA and ApoC and ApoE, and we know what they do and why they are here. And it has a phospholipid, mostly phosphatidylcholine, PC. So what it does, once it's in the blood, in the circulation, it will take up cholesterol from all peripheral tissues. As it takes cholesterol up in the form of free cholesterol, it immediately esterifies by LCAT, as we said. And we said LCAT needs ApoA1 for activation. So ApoA1 activates LCAT, which is bound to the nascent HDL. Now, LCAT will start to form these cholesterol esters, transferring fatty acid from our phospholipid, phosphatidylcholine, to cholesterol to form the more hydrophobic cholesterol esters. Being hydrophobic, they will be sequestered in the core of the lipoprotein particle here, HDL. طيب, as you would imagine, HDL as it circulates and collects cholesterol esters, they might get overloaded or oversaturated, and with too much cholesterol esters, that might inhibit the enzyme, the LCAT, the esterifying enzyme. So how can we go about this? There is an enzyme, CETP, cholesterol ester transfer protein. So in the circulation, there will be an exchange of lipids between HDL and VLDL. HDL will give VLDL some of its cholesterol esters in exchange for triacylglycerol عشان يقلل الكوليسترول ester content of HDL so it can collect more. Release the inhibition from the LCAT, the esterifying enzyme, so we can take up more and more cholesterol esters. And we know eventually VLDL, where will it go? Back to the liver. So it helps HDL with its function. يعني الكوليسترول الجود cholesterol that's going back to the liver, even if it's within VLDL, it will reach the liver eventually. طيب, we have قلنا the nascent HDL, which is discoidal. It's sort of protein and phospholipid. As it goes, it collects cholesterol ester and it becomes slightly spherical shaped with some cholesterol ester content, but it's relatively poor, and that's what we call HDL3. It keeps circulating, collecting more and more cholesterol esters, and transforms into HDL2 eventually, which is more cholesterol ester rich. That will go to the liver to deliver its cholesterol esters to the liver. So that's the pathway. We have our discoidal nascent HDL with ApoA1. No yellow here because it does not have any triacylglycerides. Synthesized in the intestine and in the liver, released as it goes in the circulation, receives cholesterol from the periphery, which is esterified by LCAT into cholesterol esters, sequestered in the core here. The esterification is, as we said, catalyzed by LCAT, activated by ApoA1, and the acyl groups are provided for esterification. Fatty acyl groups are provided by phosphatidylcholine. So that's our HDL3. With further cholesterol ester uptake, we will have HDL2. Because of the higher content of cholesterol esters here, we need to transfer some to VLDL. So here is the exchange with CETP, and hepatic lipase also release some of the triacylglycerol. طيب, by now you know the functions of HDL. So it has two apolipoproteins. It keeps them as a circulating reservoir so that it can give them to other particles. ApoC2 goes to VLDL and chylomicron to activate LPL. And ApoE also it's given to them, and it works as a receptor for receptor-mediated endocytosis of the remnant particles. It also functions to take up unesterified cholesterols from the peripheral organs and deliver them to the liver. It also has an important function in cholesterol esterification because it takes up free cholesterol from periphery and converts it to cholesterol esters. Lastly, it functions significantly for in the reverse cholesterol transport from the periphery to HDL to the liver and steroidogenic cells, which can or need to utilize this cholesterol. Right. We have heard of the good cholesterol carrier designation of HDL because of the inverse correlation or relationship between the levels of plasma HDL and the risk of atherosclerosis. So it's the good cholesterol carrier. There are factors that influence the levels of HDL in the circulation, increasing them, which is beneficial, obviously. Exercise, that's why people with risk to relieve the risk of cardiovascular disease are advised to exercise more to increase the levels of HDL. Also, estrogen administration, that's why women have less risk of cardiovascular disease before menopause, and the risk increases after menopause, and sometimes they are prescribed with estrogen replacement therapy after menopause to reduce the risk. However, recent evidence showed, it's not it can increase the estrogen replacement therapy can increase the risk of vascular disease. So this point must be utilized carefully. Also, moderate alcohol intake can increase the levels of HDL and have a protective effect, but 2 points, very small amounts are beneficial. And the other thing that it is thought that the benefit comes from the other components of the alcoholic beverage rather than ethanol itself. Uh, the risk of atherosclerosis, you must have heard of the oxidized lipoprotein particles, these small dense lipoprotein particles, and how they can predispose to atherosclerosis. Let's have a look at how this happens. So, when there is endothelial injury caused at least by oxidized LDL, we will have our monocytes gathering here, adhering to this endothelial cell and then move to the subendothelium or the antenna. They are then here transformed into macrophages, and the macrophages will consume more and more of these modified or oxidized LDL particles. So, because they are small, the smaller the LDL particles, the higher the risk because they can penetrate easily and induce the damage. The more oxidized they are, the easier they can be taken up by macrophages because this uptake process is unregulated, less specific, and with low affinity compared to the regulated LDL receptor uptake. And the denser the LDL particle again increases its plasma half-life. So these three factors increases the risk of plaque formation and atherosclerosis. So now we have our macrophage. It can take up these oxidized LDL particles, and as these are taken up, it will transform into foam cells. These foam cells will start to accumulate and form the result in the plaque formation. They release growth factors and cytokines, in stimulating muscle cells also to transform into foam cells. As they gather these foam cells, they will block the capillary and or the blood vessel and cause plaque formation. They will eventually rupture and result in clots. And as you can see here, the comparison between the scavenger receptors on macrophages, which take up the oxidized LDL, which is modified compared to the regular or the normal LDL particle. So they have low affinity, but they are highly non-specific and non-regulated, so they can take up as much as possible of the LDL particles without regulation. It's unregulated. Unlike the LDL receptors, which are specific and have high affinity and are regulated. So when there is sufficient cholesterol, they will be down-regulated and stop cholesterol uptake. That's why we need antioxidants. The presence of antioxidants will prevent the formation of these oxidized LDL particles, which are atherogenic compared to normal LDL. Right, there is another lipoprotein particle called lipoprotein A. It is exactly the same as LDL, similar to LDL. The only difference is that it has an additional apolipoprotein called apolipoprotein A. The higher the plasma levels of this lipoprotein A, the increased the risk of coronary heart disease. Why is that? Let's look at the difference between the only different thing between lipoprotein A and LDL is our apolipoprotein A. It is structurally similar to plasminogen, the same as plasminogen in structure. So it will compete with plasminogen. Plasminogen is a precursor protease that breaks down the fibrin clot. It binds to the fibrin clot and breaks it down. So it's important to prevent blood clots. So when we have a compound that is structurally similar to plasminogen, it will compete. So when it competes, this ApoA competes with plasminogen for binding to fibrin. It will bind and inhibit fibrinolysis that can be caused by plasminogen. So it predisposes to thrombus formation and the heart attacks. Okay. طيب, these are extra bits that are just to give you a feel of clinical relevance of these lipoproteins and just lipoproteinemias, medical clinical conditions that are due to defects in these lipoproteins. So we'll have total 5 examples, just briefly. In general, this class of diseases or defects are called dyslipoproteinemia. So there is something wrong with this, whether high levels or low levels, as long as they are associated with disease eventually. So either high LDL or high high chylomicron, or what's in low level is less beneficial, HDL. If it's low, that's kind of harmful. So either high LDL, high VLDL, or high chylomicron, high cholesterol, or low HDL. So we have 2 classes of this lipoproteinemias, hypolipoproteinemias or hyper. Let's start with the hypolipoproteinemias. When are they harmful? When there are low levels of HDL. We have Tangier disease, an example. Tangier disease is extremely rare. It's a deficiency in ABC1, ATP binding cassette A1. The function of this ABCA1, we did not talk about it, but its function is to remove these free cholesterol from the peripheral tissues to HDL. So it helps in the transport of cholesterol from the organs to HDL. So with no ABCA1, there is no HDL because it's not assembled. وش اللي عندنا بيكون? We will have ApoA1 standing on its own. It's not loaded with cholesterol esters. فbeing lipid poor, only empty shell اللي هي ApoA, it will be degraded. So we will have no HDL, and that's Tangier disease. One of the important clinical features of Tangier disease is the enlarged tonsils. طيب, beta lipoproteinemia, it has another name, Korn-Zweg disease. It's rare again, and its deficiency in MTP, microsomal transport protein اللي تكلمنا عنه في الأول. Its function is to load الApoB lipoproteins with lipids. VLDL والchylomicron هم الApoB lipoproteins that are needed to be loaded with lipids when they are synthesized. صح? Chylomicron in the intestine needs to be loaded with dietary lipids. VLDL in the liver needs to be loaded with endogenous liver synthesized lipids before they are released. So with defective MTP, they cannot be loaded with lipids. So these particles, VLDL and chylomicron, are not formed properly, only a small amount is formed. And these triacylglycerols that are supposed to be loaded into these particles will remain where they are. They will accumulate in the liver and in the intestine. In order to memorize the name, maybe the A is for no beta lipoproteinemia اللي هم lipoproteins اللي عندهم الApoB اللي هم chylomicron and VLDL. طيب. Hyperlipoproteinemia, when there is increased level of one or more of the lipoproteins, it can be primary, as we have seen, caused by genetic defects. These genetic defects or mutation can affect the production of these lipoproteins, increase their production, or impair their processing, so they will accumulate, or also impair their cellular uptake, so cells will accumulate in the circulation, or cause defective removal as well by one mechanism or another. Secondary causes of hyperlipoproteinemia can be attributed to other conditions such as obesity, diabetes, hypothyroidism, renal disease, liver disease. Um, excuse me. Excuse me. Other lifestyle factors such as diet, drugs, also alcohol intake can cause hyperlipoproteinemia. Remember the exogenous pathway of chylomicron? Imagine if we have no LPL, no lipoprotein lipase enzyme defective, or its activator is defective. One of these two mechanisms. The outcome is the same. No, chylomicron cannot release its triacylglycerol, so we will have increased chylomicrons in the circulation and increased triacylglycerides because they are the component that is not degraded, that is not released. And this is what we call hyperchylomicronemia. Hyperchylomicronemia في high chylomicron, high triacylglycerols, and the mechanism or the cause is defective LPL or ApoC2. Another type is familial hypercholesterolemia. So I would, as you would imagine, since it's hypercholesterolemia, it will be relevant to LDL, and the precise defect is in LDL receptor. So the circulating LDL cannot be removed, and it will remain in the blood with elevated LDL and therefore elevated cholesterol because it's the main component of LDL. It's quite common and it's genetic, 1 in 500 of the general population. It could also be due to other reasons. مو بس ال LDL receptor هو اللي defective. ممكن ال ApoB100 اللي هو في ال LDL particle، يعني يا اما receptor يا اما recognition recognition particle on the lipoprotein. If there is defective ApoB100, it will affect its binding, make it bind poorly to the receptor. بس the more common cause اللي هو FH, familial hypercholesterolemia, is LDL receptor defect. Lastly, we have this beta lipoproteinemia or broad beta disease. So it has to do with the beta or the ApoB lipoproteins اللي هم الكايلومايكرون وال VLDL. This disease is, to help you memorize it or understand it, this beta or broad beta اربطوها بالرemnant. This is, you can call it the remnant disease because it affects the clearance or the removal of the remnant particles from the circulation. ايش هم الرemnants؟ الكايلومايكرون remnant وال IDL. IDL هو ال VLDL remnant. So there is defective clearance of these particles. ايش المشكلة؟ هل هي في الريسبتور ولا في البارتيكل on the lipoprotein؟ المشكلة هنا في الأبو E اللي هو البارتيكل الأبوبروتين اللي على السيرفيس that is recognized by the receptor. فعندهم impaired binding to the receptors، وبالتالي إيش الأوتكم؟ إنه increased remnant particles in the circulation. Let's just discuss few isoforms of this ApoE. ApoE has 3 isoforms: E2, the least common and the poorest, with the poorest binding capacity. There is APO3, more common, and there is APO4. APO2 being of the poorest binding capacity, people who are homozygous for this APOE2 show the lowest binding capacity of the remnant particles to their liver receptors. So they will have increased levels of remnant particles in the blood with a high hypercholesterolemia. So here is the defect in the ApoE itself. Okay. So that concludes our session. I hope it was not repetitive and it furthers your knowledge on this topic and help you understand some of few of the things that you did not understand in the past, added to your knowledge. Again, if you have any questions, please drop me an email, uh, and feel free to ask questions at the moment if you feel like it. Right. Since it appears there are no questions, I'll conclude here. We have still one more session, inshallah, next week, الاثنين. I would like to thank you. Wish you all the best إن شاء الله في الاختبار. Again, هذه السيشن داخلة في الكويز الثاني وداخلة في الفاينل. فإن شاء الله we will have more time after the first quiz to address any questions you have.